Hello there. This video is going to explain 3 new patterns of inheritance: incomplete dominance, codominance, and sex-linked traits. At this point in time, you already know how to do complete dominant problems. These are problems where there are only 2 phenotypes present. This is what we've been doing all along, where a key would look like big R is red and little r is white. Okay, today we're going to learn about situations where we have 3 different phenotypes. So we're moving over here into the middle area of the flowchart. If the third phenotype is a blend of the dominant and recessive, this is a situation of incomplete dominance. Your key is going to have to look like this. You must list out all 3 phenotypes and all 3 genotypes, because now the dominant allele is incompletely dominant to the recessive. So the third phenotype is a blend, all right? So the heterozygote is going to show the blend. Okay, we're going to do a practice problem. If you flip to page 6 in your packet, it will look like this. So they tell you, in Andalusian fowl, a breed of bird, the gene for black plumage is incompletely dominant to the gene for white plumage. The heterozygotes have blue plumage. So the first step is to make your key. All right, so 2 big B's is going to be black. 2 little b's is white. And then the heterozygote, big B, little b, is going to be blue. From there, the problem is exactly the same as what we did before. So they want us to cross a black bird with a blue bird. Then you fill out your Punnett square just like before.
Is that for class?
And then they wanted the expected genotypic and phenotypic ratios, but for here, I just asked the class to tell me the percent of blue offspring, and in this case, that would be 50% blue, because these organisms right here are going to be the blue ones. So basically, the only difference here is that you have to write your key in a very specific way. You must list out all 3 genotypes and all 3 phenotypes in order to get all of the points. Okay, if you flip back to the front page of the packet, back to the flowchart, so we've talked about incomplete dominance. Now we're going to talk about codominance. Codominance is a situation in which you're going to see things like stripes or spots as part of the phenotypes, right? The root co means both. So both alleles for this trait are dominant. In this case, that means that when you look at the key, you're going to see all capital letters, all right? And in fact, 2 different letters, all right? So this might be, for example, in flowers, we might have red is codominant to white, and the heterozygote is going to show red and white stripes. Okay, we're going to do a practice problem. Okay, if you could flip to page 8 in your packet. It says at the top here that the genetic disease sickle cell anemia is caused by 2 codominant alleles. 2 normal genes produce normal red blood cells, and 2 sickle cell genes, capital S, cause the disease to occur. Individuals afflicted with this disease have sickled red blood cells that are unable to carry oxygen properly. The heterozygous condition is not fatal and seems to offer protection against malaria. So the first step here is to write out our key. So they told us that individuals with 2 capital N's are normal. 2 big S's have sickle cell anemia, and then the heterozygote are going to be normal. And they're protected against malaria. Okay, so it says in the problem, if a man and woman are tested and found to be carriers of the sickle allele, what is the probability that they will have a child who has sickle cell anemia? New word here, carrier, is a synonym for heterozygous.
Am I supposed to do something?
Okay, so carrier means heterozygous. All right, so if a man and a woman are both carriers, all right, there's your cross. Fill out your Punnett square just as you normally would. All right, and the probability that they will have a child who has sickle cell anemia would be 25% sickle cell anemia. All right, so basically, again, just like incomplete dominance, it's all about the key. You have to make sure you list out all 3 genotypes and all 3 phenotypes, and in this case, all the genotypes have to have uppercase letters because all of the alleles are dominant. Okay, the last topic we're going to talk about today is sex-linked traits. So if you could turn to page 23 in your packet, we're going to do a couple of practice problems here, and then we will do a sex-linked pedigree. All right, if you read the top, it says, this situation involves the sex chromosomes, pair number 23 in humans. Pair number 23 in women consists of 2 X chromosomes. Males have 1 X and 1 Y chromosome. Sex-linked genes are carried on the X chromosome. In humans, sex-linked refers to X-linked. The Y chromosome contains genes for male characteristics and determines the sex of the individual. Hereditary illnesses such as color blindness and hemophilia are carried on the X chromosome, and most sex-linked diseases are recessive. Okay? Because males have an X and a Y chromosome, any recessive disease gene on the X chromosome will show itself. A woman has 2 X chromosomes and may carry or hide a sex-linked recessive gene if her other X chromosome is normal. A woman who is heterozygous for a sex-linked gene is termed a carrier, which we talked about in our last problem when we did the codominant problem. Males cannot be carriers of sex-linked traits. They either show the trait or they don't. So for these problems, again, your key is very important. You must make sure when you're doing a sex-linked problem that your key has to have X's, and then you must have superscripts on your X's. So in this case, they've done X big N. All right, so it might be hard to see, but this right here is an X big N, and this is an X little n. Okay, so you need to remember that color blindness and hemophilia are sex-linked recessive, so X big N is normal, X little n is colorblind. The problem says, what is the probability of children being colorblind if a normal vision man marries a woman who is carrier for colorblindness? Now it matters the man and the woman. All right, so a normal vision man, he's a man, so he has X and Y chromosomes. He is normal vision, so he has a big N on the X. We don't put anything on the Y ever. He is going to be crossed with a woman who is a carrier for color blindness. If you recall, carrier means heterozygous, so she's a woman. She has 2 X's, okay, and she's a carrier, heterozygous, so X big N, X little n. Then you fill out your Punnett square as usual. Okay, so when you fill out your Punnett square, I put the male across the top, the female along the side. You'll see that the probability of getting kids who are colorblind is going to be 25%. 25% colorblind. Okay, if they ask you the sex of that child, you would say a boy, because it's X little n, Y. If they ask you the chance of getting a colorblind son, then you would tell me 50%, because of the sons here, 1 out of 2 of them would be colorblind. All right, now if you flip to page 28 in your packet, we are going to do a sex-linked recessive pedigree. So this pedigree on page 28 follows the trait of colorblindness, which we know is sex-linked recessive. So first, make sure you have your key. So X will do big A is normal, and X little a is colorblind. Okay, the first people to do on a sex-linked pedigree, again, are all of the shaded people. We know that they are colorblind. Now circles and squares matter. So this individual right here is a colorblind male. We know he's male, he's a square, we know he's colorblind, so he is X little a y. So all of the shaded males are X little a y. The female down on the bottom, she is a colorblind female, so she must be X little a, X little a. The next people you want to do are all of the unshaded males. They're all normal males. So all of the unshaded males get X big A, Y. Okay. All of the unshaded females, we know for sure they have X big A. We can't be certain what that second allele is yet, so we need to go through and make them all X big A, X blank. And then the last step is to go through and figure out if we can fill in any of the blanks, just like we did with the other pedigrees. So for this female, all right, you would ask yourself, do I know this person's parents? No. Do I know her offspring? Yes. Here they are down here. Do they have any offspring that are shaded in? Yes. These two. That means that she must have a little a on this line because both of these offspring are going to get their Y's from their father, all right, which means mom had to give the X little a. Okay, so we do the same process for the rest of the females. Do we know her parents? Yes. Are either of them shaded? No. Do we know this person's offspring? No. So we leave it blank. Do we know the parents? Yes. Neither are shaded, and we don't know their offspring, so we leave it blank. We don't know this person's parents. We do know their offspring. She does have shaded children, so this must be a little a. For this individual, neither of her parents are shaded, but she does have a shaded daughter, so that means she must have a little a on the line. And last but not least, this individual over here, her parents are not shaded. She doesn't have any kids, so we don't know enough, so we leave it blank. Okay? And then you would be done. All right, so after we went through all those practice problems together as a class, the homework that you need to work on are pages 9, 12, 25, 26, 27, just number 8 on page 27, and just number 2 on page 28. Page 9 and 12, these are going to be practicing incomplete dominance and codominance problems. 25, 26, 27, 28, these are going to be practicing sex-linked problems and specifically sex-linked pedigrees on 27 and 28.